Very big welcome to the authors of an intercompany perspective on practical experiences of predicting, optimizing and analyzing high concentration biologic therapeutic formulations. I'd like to welcome Preeti Desai from BMS and Patrick Garidel from Boehringer Ingelheim, Francisca Gbormittah from GSK, Kamen from Regeneron, Brittney Mills from AbbVie, and Nachu from Merck, Shubhadra from GSK, and I'm Elaine, you know me. Erica sadly can't join us today. Okay, so what we're going to do today is we're going to talk you through a publication which will very soon be resubmitted to JPharmSci. And this is a reflection on a set of surveys and discussions that took place over several years in the BioPhorum formulation and drug product developments workstream. We'll start off with an introduction, and then we're going to cover the key areas relating to this. There's solubility, viscosity, stability, analytical testing considerations, then some approaches for delivering large volume doses. We'll finish up with some conclusions and acknowledgements before we move into our Q&A session. Okay. So this presentation is a composite view of opinions, and it's shared by the whole of the BioPhorum development group work stream. And it shouldn't actually be attributed to the individual positions of the participating companies. The contributions have come from a wide range of companies within the development group formulation work stream. And they have so many different product portfolios, business models, that we think it is really a representative summary of overall industry practices. So I'm now going to invite Doug to take the place as presenter. Doug is from Regeneron to talk us through the introduction. I'm going to pop my camera off, so just the presenter will stay on there. Over to you,
Thanks
Doug.
A lot. Thanks a lot, Elaine. Let me just start by thanking everybody for your attention and for your interest. It's really great to be here. And it was really great to be part of this team and this survey and author on this paper. This paper was really an amazing collaboration based on surveys conducted within the formulation and DP workstream, mainly in 2020, as an effort to gain a perspective among pharmaceutical companies, our collective experience on predicting, optimizing, and analyzing high concentration biologic formulations. So what's the problem we're trying to address? There's a trend, as we're seeing today, towards increasing demand from clinical teams to develop higher concentration biologic formulations to deliver higher doses convenient for patients. These subcutaneous drug products are not only more convenient for patients, they can improve adherence to treatment regimens, lower treatment costs, and provide a competitive advantage to the companies that develop them. Additionally, high concentrations can be beneficial from a logistic perspective, as this can reduce things like volumes and improve frozen storage capacity, shipping, and handling during manufacturing. Some minor things, but really, really helpful. The responsibility of developing these higher dose formulations falls on formulation and DP development teams. So this is certainly an appropriate forum to hash out these issues. So what are some of the challenges associated with high concentration and high dose formulations? Well, for one thing, as protein concentrations increase, solubility approaches upper limits. Stability decreases and viscosity increases. We tend to have limited amounts of material early in development, and we always have limited human resources. Analytics can be challenging due to the need for dilutions and to stay within the dynamic range of instruments. So these and other challenges can lead us to ask, can we utilize in silico or biophysical tools to more efficiently model or predict stability and solution behavior? And how successfully have these methods been applied to real development? So what did we learn? Well, first and foremost, we gained an industry perspective on practices for developing high concentration formulations. We got a good understanding of the usefulness of predictive tools and models, which may surprise some people. And I think we're also learning some new things about models and predictions that we may not have covered in this paper. We discussed various approaches to these, so things to like approaches to assess solubility, viscosity, and stability, and learned how others approach these challenges. We also learned important points on sample preparation and analytical testing. Finally, we touch on technology for delivering large doses in lieu of high concentrations, as it's not always possible to get the desired dose in a low volume. Can we go to the next slide, please? So here's a snapshot of the manuscript. This has not been published yet, but it's been submitted to the Journal of Pharmaceutical Sciences. We received a number of comments from the referees. All the comments have been addressed, and the revised manuscript is going through legal review and approval from the various member companies. We anticipate legal approval in the next, I guess, week or so, and hopefully we'll resubmit it soon after. So be on the lookout for that. Hopefully you'll find this interesting to read as it was to write. All right, and I think next slide, and we'll get into the first section of the survey. So the first section is solubility, and I'd like to hand this off to Patrick.
Thank you, Doug. Okay, the first section and question will relate to everything to solubility. So the next slide shows a little bit what came up. It was clear with these drugs that we have, these proteins that we have, and especially if you go for subcutaneous application, high dosages needed. And with this, of course, high dosages mean to the limitation of the, let's say, 1 milliliter subcutaneous application, we need also high solubilities, meaning high protein concentration going on with high solubilities. The survey feedback was also that solubility is a fundamental property that affects, of course, the colloidal stability of such formulation, high concentrated formulation, and these are, of course, defined by the physical-chemical properties of the API itself. Of course, the solubility depends on the molecular properties themselves, but also you can modulate it by changing formulation conditions. Questions are also related to different strategies around the solubility issues. The first one was really how to define solubility. And it came up from the survey that it's quite difficult to define solubility itself. And the reason for this is the fact, for example, that these high concentrated protein solutions may form metastable phases. They can phase separate, they can gel, they can, of course, increase viscosity, as heard this morning, and sadly also form protein particles that are often irreversible. So this was the thing that's difficult to define where is the solubility exactly. As a consequence of this, the feedback from the survey came up that different surrogate parameters were used to somehow define solubility, for example, using a precipitation, extrapolation, for example, with PEC, and then define solubility, or to concentrate up your protein solution and use turbidity or a certain protein concentration, a particle appearance, particular form, and somehow to define there a certain limit of protein solubility. Of course, protein solubility is strongly temperature dependent, so we have also to be careful about temperature. A question was also related to the solubility, the thermodynamic versus kinetic solubility that can be measured. And it came up that it is basically difficult to measure the thermodynamic solubility for these larger molecules that we have. And it was also mentioned that for smaller molecules like peptide is much easier accessible. Half of the respondents, namely 7 out of 14, mentioned directly that they don't measure thermodynamic versus kinetic solubility at all. So this was the feedback. On the next slide, it was an information related, and this is quite important, namely, does solubility impair your development program at all? And 8 out of 15 respondents mentioned that they face situations where protein solubility has limited the dose required to be delivered for the clinic. So basically, this is definitely a strong outcome of this also. We asked also the folks about how to measure solubility, how to appropriate this. And there basically was 3 big feedbacks. The first one, of course, to use the UFDF, concentrating up the protein solution to a certain concentration, and use this as a method to measure the solubility. Another method was mentioned as precipitation, whether use ammonium sulfate or PEG precipitation. However, for these 6 companies discussed this. However, they mentioned that using this assay, it is just used a comparative approach. So basically, you check different formulation, you measure it, and then basically make a kind of ranking, but you cannot really make an absolute solubility limit, so you can reach X gram per liter, so this was not it. And also one feedback mentions that they use a solid protein and then redissolve it, and with this also try to measure the protein solubility. The strategies to improve protein solubility were first to optimize the formulation pH or buffer, 7 feedbacks on this, and also on the other side to use excipient screening. 8 people asked, or 8 companies also mentioned to use this excipient screening in order to trying to improve the protein solubility. It was also proposed in the survey to use lower concentration formulation and even by one participant to redesign the molecule itself. One remark in the survey was also that the bulk drug substance need to reach even higher protein concentration compared to what is needed for the drug product formulation itself. And they also recommend strongly that the people from the drug product side have a strong interaction with the drug substance colleague in order to learn already from these people how to make, let's say, and to improve the protein solubility. Besides this, viscosity is also quite important, Shubhadra. What was the fact about this?
Thanks, Patrick. So I will cover viscosity survey questions. Yes, so we all aware of that viscosity is one of the major challenge and a common challenge in developing high concentration subcutaneous drug product. So I will start with the right hand graph. So we asked the question about the viscosity limit, and we see that 5 of respondent indicated that 20 centipoise is the limit, and then 5 said 30 centipoise is the limit. So basically there is a tie between 20 and 30 centipoise. Next, we asked about the questions on predictive tools. So 4 of 15 respondents indicated that they use in silico tools to understand viscosity, and in some cases predict viscosity. And the tools that have been used are machine learning and molecular models. And next was biophysical methods. So 7 of 15 respondents indicated that they are using biophysical methods for understanding or predicting viscosity at higher concentrations. So for biophysical tools, they are using dynamic light scattering, static light scattering, QCM, which is rheological parameters, G prime and G double prime parameters, and then zeta potential, which is the charge of your protein under experimental conditions. And our next question was about the experimental methods companies are using, and everyone, 13 of 13 respondents indicated that they are screening for pH, buffer, and excipient to decide about the formulation with lowest viscosity. And then we also asked for excipient, which is the most popular or mostly used to reduce viscosity, and arginine was the winner. Everyone is using arginine to reduce viscosity. Our next question was regarding other strategies to overcome viscosity challenges. So 16 of 13 respondents indicated that they use a delivery device devices optimization, so patch pump by decreasing the flow rate, or auto injectors with different strength of spring. So these are delivery device strategies. And then 4 of 13 respondents indicated that they use a primary container closure system to optimize the viscosity. So basically syringe barrel optimization of silicon level within syringe, stopper geometry, and stopper siliconization, needle optimization, thin wall needle, changing the gauge of needle, that means larger gauge needle, and increasing the diameter of needle. So these were the strategies. For viscosities, yeah, next is stability. So I'll hand it over to Doug.
Okay, thank you very much. So stability, we're going to split this up amongst the 4 of us. So we'll each get a little bit of a portion of this section to talk about. If you go to the next slide, please. Thanks. All right, so I think I think the major stability obstacle to overcome in high concentration formulations is the increase in aggregation, such as soluble high molecular weight species or insoluble aggregates and particles. And this is a function of increasing protein concentration. I think that's a fairly safe thing to say. There's other things to think about, but aggregation is something that comes to mind. So in the paper, we review some high-level approaches to decreasing aggregation in high concentration formulations, including the introduction of stabilizers into the formulation, such as sucrose and other sugars, amino acids such as arginine, surfactants, or salts. We also mentioned that lyophilization is a common approach to stabilize proteins. This approach is becoming less and less favorable, though, due to commercial and patient convenience reasons, and liquid formulations are becoming much more desirable. So we get into the survey. Survey respondents were queried on practices for selecting high concentration formulations with stability as the factor in formulation decisions. So this is specifically focusing on stability. So it's pretty common to assess stability of therapeutic proteins at accelerated and stress temperatures and conditions. And it turns out that 15 of 15 respondents use accelerator stress stability results to select high concentration formulations. 9 indicated that stress data either sometimes correlates, or correlates to some degree to long-term stability. We also are showing some of the comments from the respondents on stress stability studies. So, for example, stress stability study. One respondent said that stress stability can accurately predict chemical stability, but thermal stress data, for example, the 40C conditions, may exaggerate the amount of degradation, especially for physical stability, referring for the most part to aggregation. The relative performance of formulations can be predicted, and top candidates can be selected for additional long-term studies, but absolute performance is difficult to predict from stress stability. So these studies are good for comparing different formulation conditions. 4 respondents indicated that stress and accelerated data did not correlate to long-term stability, though 2 agreed that the conditions were useful for ranking candidates or comparing formulations, similar to the previous comment. So this is referring to the figure. When asked which prediction approach do you rely on to select the formulation so for further long-term assessment, so this is starting out and then what you can bring to look at further long-term assessments. 13 of 15 respondents did indicate stress stability was their preferred method. 7 of 15 said biophysical, and 1 said in silico. In a follow-up question about temperatures used for accelerated or stress studies, most respondents use 25 and 40 C, but others such as 30, 37, or even 45 C are used. Of note are some comments, and those are listed on the slide. Respondents indicated that the choice of accelerated or stress temperature depends on the T onset, the TM, or the aggregation temperature. One respondent specifically indicated that 37 C would be used as a stress temperature if the TM or aggregation temperature were relatively low. Another indicated that a temperature of 10 to 15 degrees C lower than the T onset would be used as their stress temperature or accelerated temperature. Okay, next slide, please. And Shubhadra is going to talk about the reliance on tools to predict stability.
Thanks, Doug. So previously we have discussed tools for viscosity. Now on this slide, I'm showing you the survey results for stability predictions. So on the top pie chart, we are showing the response for in silico tools. So here 11 or 15 respondents said that they are not using in silico tools. And 2 respondents said that they are using in silico tools, and 2 said that they are using it sometimes. So majority people said that they are not using in silico tools for viscosity predictions. Then next is about biophysical methods. So 11 respondents indicated that they are using biophysical methods to indicate or predict formulation stability. 2 respondents indicated that they are using it sometimes, and 2 are not using. So based on this, we can say that biophysical tools are typically used to understand stability, and in some cases correlate with stability data. Yeah, we can go to next slide. And here I'm talking about biophysical tools for thermodynamic stability and colloidal stability. So on the left hand side, it's for thermodynamics stability. So 14 respondents indicated that they are using a DSC to understand and predict stability. Structural stability, and 12 respondents indicated that they are using fluorescence based technique, typically DSF technique for determining TM or aggregation temperature. On the right hand side, I'm showing you the colloidal stability tools. The first one is self-interaction nanoparticle spectroscopy. So 3 respondents indicated that they are using this technology, and recently a paper has been published where they have used this technology to understand interactions of co-formulated maps. Yeah, but yeah, limited understanding or limited experience on that technology. Then dynamic light scattering, which is a high throughput technology, and 15 respondents have indicated that they have used this technology. And the static light scattering, 10 respondents indicated that they have used this technology. So most people are using DLS to understand colloidal stability. And again, the data sometimes correlates with real-time stability, sometimes does not. So majority of people are saying that they do real-time stability to show that a given drug product at a given high concentration and in final container closure can be advanced to late stage clinical or commercial space. Yeah, so yeah, that's this biophysical tools section. We can proceed to Francesca's section.
Thank you, Shubhadra. So in this piece, we had 3 questions relating to other stability considerations that most companies would evaluate regarding stability. For these 3 questions, I'm going to start from the left-hand side. The first question was relating to silicone oil presence. So there are a number of publications actually that discusses the potential impacts of silicone oil, and one of them is immunogenic impacts, as well as aggregation induction due to the presence of silicone oil. And so in this survey, we wanted to just get practical understanding from companies that participated in the survey. And the simple question was, have you observed any impact on your stability that relates to silicone oil? The responses, as you can see from the pie charts, were split. Half of the respondents indicated that they have observed impact on their stability, which was due to the silicone oil presence, while the other half indicated that they did not observe such impacts relating to silicone oil. One respondent did make a note that they did an investigation because they did noted that the stability was due to the stability issues they were having was due to the presence of silicone oil. And their investigation led to the fact that they could correlate increases in the particulates that they were seeing, both visible and subvisible, was related to the silicone oil that they had in the product. And they mitigated this observation by simply adding some amount of surfactant to their formulation. The second question in this particular section was relating to, on the right-hand side, pre-filled syringes. So for high concentrated products, mostly pre-filled syringes are evaluated as the container closure or one of the container closures. So we wanted to understand if there are differences in the panel of testing that companies utilize to test the stability of these products. And the responses did indicate that most companies' first approach is to run the same type of tests or studies as they would typically do for bioproducts. And then for PFS, they do have additional specific evaluations, which we have listed here. And those included silicone and tungsten compatibility evaluations, injection forces studies, syringe orientations, vaporized peroxide compatibility studies, shear stress through the needle studies, as well as evaluating the deliverable volume. The last question we asked was relating to companies' approaches towards late phase stability challenges. So as we know, if you have a late phase stability challenges, it may lead to maybe significant delays in the launch of your program, or even in certain worst case scenarios, it may cause the program to be canceled entirely. So again, our question was asking industry typically what is the current approach they are using, and the responses from the survey show that most companies evaluate these challenges by first looking at the failure mode. And depending on the failure mode, there are a variety of ways that they would address the concern. And as listed here, some people mentioned that they would look at the formulation to redevelop it. Others suggested that they will look at a presentation change. So for example, if they're seeing stability issues with a liquid formulation, they may decide to go to a frozen or a lyo formulation. Some people indicated that they would step back to look at their manufacturing process. Maybe it's impurities coming from the manufacturing process, and so they may be able to optimize from there. Others mentioned reducing shelf life. So for example, if the target shelf life was 24 months and you're having issues after 18 months, you may consider reducing it to 12 months, depending, of course, on the program goal and indication that you are trying to target for. Others would look at container closure system. And the two last comments that other people provided included pausing clinical trials, as well as recalling product. Can we go to the next slide, please? I'll now hand it over to Nachu to continue with the stability discussion.
Thank you, Francisca. As you can see so far, the stability piece of the survey has been pretty detailed and inclusive. And so I'll drive home the last section of the stability challenges that is required to be addressed for the high concentration formulations of biologics. And for complete development of high concentrated formulation development, we need to understand the biochemical and biophysical stability, compatibility with the container closure system, viscosity profiles, and device functionality. While stage appropriately, people may focus on some of these elements, but for complete product development, all of these elements need to be fully understood before coming up with a robust drug product. And so the first thing we queried was the challenges due to the drug substance availability. How do companies deal with the material requirements, right? And so the response we got was 11 out of 17 folks indicated that formulation selection is rarely determined using stability data generated with solutions at lower concentrations. So meaning that people tend to work at high concentrations close to the target as possible, and some do consider bracketing approaches. So where they may be looking at lower concentrations and then a high concentration, and then try to come up with a stability indication with the available material. While this may be suitable for early stage development, but for the late stage, we need to study the stability at the target concentration. Next, we asked about the challenges that are coming from the excipient acceptability. Do they have to, what do they have to do? For this question, the member companies, 12 out of 16 of them responded strongly that they strongly preferred excipients currently used in commercial products. And so they want to be in the safe zone, so where something has already been established in the market. While 10 out of 16 respondents rely solely on, generally regarded as safe excipients. And so again, this has a lot of safety built into that. And so that is the approach for 10 of those respondents. Interestingly, 8 out of 16 respondents indicated a willingness to explore novel excipients with available safety data. What this means is that monographs have not been established for these novel excipients, and so they need to be tested as such. But you do have some safety data available. But a few respondents, 2 out of 16 respondents did indicate that they're interested in establishing novel excipients. And so they don't mind introducing a novel excipient and then generate the appropriate safety data, if you will, and then decide their formulation for the high concentration drug products. So it's an interesting array of questions on stability, and we did get detailed response from all the queried companies. And so how do they go about testing this? I think that's what is going to be the subject of the next section, which Brittney will be dealing with. Study setup, execution, and reporting. Brittney, it's all yours.
Thanks, Nachu. Next slide, please. So I'm just going to give a little bit of insight into the sample preparation, analytical testing, as well as in-use considerations when it comes to looking at high concentration formulations. So first off, as Nachu kind of alluded to on the last slide, having a stabilizing excipient is generally required for high concentration formulations. In order to identify which excipient is appropriate, high throughput methods are typically used, and therefore minimal amounts of sample would be required. Companies reported that this can be performed manually or using automated methods. When it comes to analytical testing, most respondents stated that there is automation used in analytical processes. For example, viscosity, DSF, DSC, DLS, or even MFI and HPLC. When we're thinking about complications for analytical testing when it comes to high concentration formulations, a few things that were noted included that light scattering methods may be limited due to scattering artifacts. In addition, with high concentration formulations, we have to consider sample dilution and the effect of sample dilution when it comes to size exclusion chromatography, as this was specifically called out as being a concern for potentially missing any aggregates that occur upon dilution. Finally, from the analytical perspective, it was noted that new techniques may need to be developed when high concentration formulations are used and analyzed. We may not be able to simply apply all of the current methods being used to these samples. Next, I'm going to go into visual inspection and appearance. All companies, 15 of 15, reported that visual inspection is performed manually. Sometimes with high concentration formulations, it can be difficult to assess the particles if there is a significant color present in the formulation or in the sample, as well as viscosity related concerns. Camera or instrument-based methods may also be used for appearance-based assessments, as was reported by 5 of the 15 companies. As I noted before, high concentration formulations may have a more distinct color, but in general, the pharmacopoeia methods are used for the assessment of appearance. And last, to talk about the in-use considerations. When we queried the respondents, they stated 10 of the 14 stated that filtration can be used at the point of administration to remove visible particles. One respondent did note that this strategy may be only considered for early phase clinical trials. In addition, the respondents were queried on syringeability testing, and 15 of 16 companies or respondents stated that they performed this syringeability testing at temperatures representative of clinical conditions. In addition, 5 respondents confirmed that yes, numerical specifications tied to syringeability are not always in place for release and stability. Next slide, please. I'm now going to pass it on to Preeti, who will discuss the technologies that are available for delivering large volume doses when we are not able to achieve high concentration formulations. Preeti?
Thank you, Brittney, and hello, everyone. So as Brittney mentioned, I will be talking about technologies that are used across the industry for delivering large volume doses when high concentration cannot be achieved. And these will be mainly survey responses. We understood today from the presentations from various presenters that sub-Q route is favored over IV. And so we need to achieve high and perhaps even ultra high concentrations. But there is a need to maintain the plasma concentration. And so one of the factors that is important for maintaining that is controlling the amount of injection volume that can help with the rate and the extent of absorption of the proteins. So if we can go to the next slide, please. So here we queried the respondents on several or three questions that you see here. So one of them being practices for delivering technologies that were used to increase the amount of protein that were delivered. So the responses varied among different participants, and it ranged from denial of use of any technologies with no specific reference to limiting the injection volume to 1 ml. And in some cases, respondents mentioned increasing the syringe size to 2 ml. And for volumes which were greater than 2 ml, there was a preference to use pre-filled syringe devices or pre-filled syringes or devices for that matter. And respondents also proposed the use of excipients such as absorption enhancers and viscosity reducers, as well as evaluating suspensions. In addition, respondents mentioned referred to use of patch pumps and hyaluronidase co-formulation to increase the amount of drug delivered. In some cases, respondents mentioned about, one case actually, one respondent mentioned or using alternate route of administration such as IV and intratumoral. Then the respondents were also queried on technologies that were considered to administer large amounts of protein and small injection volumes. So 11 responses were received, and each response varied. And responses included use of a large volume injector, targeting higher concentration and limiting the injection volume to 1.5 ml. And then again, the respondents also suggested use of hyaluronidase co-formulation, patch pumps, or slow subcutaneous infusion. In some cases, respondents suggested the use of approaches such as nanocrystalline protein suspensions with the idea of reducing the viscosity, and then use of non-aqueous co-solvents and amorphous precipitants. And lastly, the respondents were questioned about new approaches that were used for high concentration formulation. So novel excipients were suggested by 5 of the 7 respondents, and they did mention the need for extensive work, specifically to safety assessment and requiring approval for use of those excipients, which also would, in addition to bearing the potential risk of protein instability with those excipients. And other approaches included reducing the viscosity of the high concentration formulation with use of nanoparticle formulation, crystalline formulation, and the use of arginine, sodium chloride, and other excipients that helps to minimize the protein-protein interaction. And with this, I would like to hand over the presentation to Brittney, who will summarize the conclusion from this presentation. Thank you.
Thanks, Preeti. Next slide, please. So as Preeti said, I'm going to wrap up today before our Q&A session. So first point here we have listed, the biggest challenges, and what I hope you were able to see is that achieving the high protein concentration while minimizing the viscosity and aggregation is very difficult. In addition, there is that inability to apply standard analytical methods, which may require additional method development when we are looking at high concentration formulations. Another conclusion we came to is that due to the poor correlation between the experimental stability of those high concentration formulations and the modeling, usually experimental approaches, whether that be stressed or real time, are used and relied upon to make decisions regarding formulations that will be selected. In general, physical stability at lower concentrations is not always predictive of higher concentrations, which was noted by multiple study participants. And then, as Nachu stated, there are limited, safe, and established GRAS certified excipients suitable to achieve optimal stability and viscosity. So this may be one area, for development and investigation in order to help support high concentration formulations. And then finally, there is that desire to deliver the large dose within a small volume in order to enable subcutaneous administration. It is preferred to achieve this through using excipients, as there is limited experience in application of alternative co-solvents, as well as different technologies like Preeti discussed. So with that, Elaine, I don't know if you wanted to take over for the Q&A. Acknowledgements there.
Absolutely, thanks, Brittney. So it just remains for me to acknowledge the wonderful authors of this publication who've done such a lovely job of presenting for us today, and also acknowledge people who reviewed the material for us, and particularly the whole of the BioPhorum development group, formulation and drug product workstream, you know, for completing the surveys and for the vigorous decisions that discussions that have got us to this position. So thank you very much to them as well.